we're going to go ahead and get started here because I have a lot of stuff to cover tonight and only 1 hour to do it in. So if you give me a moment here, I am going to go ahead and rearrange my boxes. And first thing I need to do is I need to make you all aware that we have a launch coming up. And I want to make sure that I get this up here so that you can make a note of it, particularly my teachers. So good morning, good evening, or good afternoon, depending on where in the world you are joining us from. So I want to first make sure that I bring your attention. So my name is Dr. Ann Weiss, and I'll give you a little bit more background on me in just a couple of minutes. But before we start the formal program, I want to make you aware that here at NASA we have a launch coming up on July 30th. I'll put it in the chat box here. It is going to be the launch of our Mars 2020 or Perseverance rover. And the launch time, as I said, it's targeted for July 30th, and it's targeted for approximately 7, all right, I think I goofed it up again, 7:54 a.m. So for my teachers, if you're looking for resources to tie in the launch of Perseverance with your curriculum this coming year, I'd like to draw your attention to the STEM toolkit that is currently on your screen with the web address there. And I'd want to make sure everyone understands that I am recording this program, and I will give you my email address so that you can contact me if you have any questions. But I'm also going to get some information from you so you can get the resources, which will include the link for this particular STEM toolkit. So I just wanted to put a plug in for Mars Perseverance, which will be launching next week, actually, a week, hopefully, from today. So hopefully, if you're planning on coming to my webinar next Thursday, we'll be able to discuss a successful launch. So this evening's program is the third in a 4-part series that I'm doing this month on human factors, and specifically tonight's program is going to focus on the nervous system. So that's going to include a couple of things. First off, I'm going to just review a few key points about the nervous system. We could probably talk an entire 16-week course about the nervous system, but I only have probably about 10-15 minutes, so I'm going to highlight just a few key things about the nervous system so that we're all on the same page. And so I ask the indulgence of our professors and our university students that just want to make sure everybody's on the same page so that when I introduce some of the research that has been done on both the space shuttle with the Space Lab and with the ISS, that'll make a little bit more sense. I'm going to talk a little bit about protective strategies based on what we've learned, and also lots of implications for Earth-based medicine. And throughout, I'm going to make sure that I include some references to instructional activities. The vast majority of folks who are on the line this evening are K-12 teachers, as I noticed by our poll. So I want to make sure that there's something in here for everyone. Chat box is going to be the best way to communicate any questions that you may have for me. I'm also going to ask if you would please, for attendance purposes, but also for resources, if you would please type 2 things. I need your email address so I can mark you as being present, and I'll explain why in just a second. And then in parentheses by your email, if you'd like the resources, just go ahead and put your email address, parentheses, resources, and that way then I can easily find the folks who want the resources. So within the next couple of days, about a week or so, I will send out an email to folks that will include a couple of things. It'll include a link for the recording of this program so you can listen to it again. It'll include a link to Dropbox where you can download my slides in PowerPoint format. It will include a PDF of the slides, and then it'll include a variety of different websites to include, for example, the Mars 2020 STEM Toolkit. So if you are for certain, please let me know your email address so that I can mark you as being present here. But also, if you want those resources, then by your email, before you hit enter, put like a little note that you want resources or something so that I know that you're interested in having that. And the reason I'm asking for the email addresses is for those of you who've been with us, but for those of you who don't know, you're able to earn a professional development certificate, particularly the teachers and the students. So I need the email address to be able to mark you as being here, because tomorrow you will get the first of 2 emails. It's the first one will be a post-event quality survey. It's a 5-minute survey, asks you to give some feedback on tonight's program. Basically, you're grading me. And then if you fill that out in about a week, which will be July 30th, you'll get a second email, which will then include a link to your certificate. And so you have to fill out the survey in order to get the certificate. And so what you'll notice, particularly for students or members of the general public, parents and stuff like that, you'll notice that the survey is geared primarily towards educators, and that's because we originally designed this webinar series for teachers, but because of the pandemic, we have opened it up to everyone, K-12 students, university students, members of the general public who are interested. So if you are interested in getting a certificate, just answer the questions the best that you can. That way then you can get the certificate, and then teachers, you can take that certificate and turn it into your credentialing office with your district or your school. Students, you can turn that into your teachers or professors as having been here. So that's why I need your email address. So if you're just joining us, please make sure you put your email in there, and if you want resources, then just put in parentheses that you want the resources, all the goodies, the copies of the stuff that I'll present tonight. I see quite a few familiar faces, but there are some new individuals, so I want to make sure that I introduce myself properly. I mentioned before, my name is Dr. Ann Weiss. I am the Educator Professional Development Specialist at NASA Langley Research Center in Hampton, Virginia. And I am originally a neuroscientist by training, so I am super excited to be able to bring this particular installment. I've enjoyed bringing the entire installment to you, but this one in particular really kind of goes after my own research interests. And so I was originally trained technically as a neuroendocrinologist, which is how hormones affect areas of the brain. And so I did that for a number of years, realized that I had the teaching, the educator passion, and so after I graduated from Arizona State, I did a pivot from the research lab into the classroom. I've taught everything from 8th grade physical science up through undergraduate human anatomy and physiology, and I've been fortunate enough to be working with NASA Education, now STEM Engagement, since 2010 on a variety of different projects. So I want to go ahead and just point that out. Now I'm getting a question from Ansh is asking, is 7:54 a.m. Pacific time or Eastern time? And I believe that is Eastern time, but I will double check that real quick. But what I want to do is first bring attention to a resource that I've kind of not had much of a chance to talk much about the last 2 installments of this particular series, and that's the Train Like an Astronaut. And this is a series of activities. It's actually a whole entire curriculum that includes things for physical education and also science activities in the classroom. So I'm going to go ahead and I'm going to actually let Paolo Nespoli, who is an Italian astronaut with the ESA, talk to you a little bit about what this program is all about. And this is also a great video to show your kids if you get them excited and want to do this curriculum with them. So stand by.
Hello everyone, I'm Astro Paolo and I'm sending you this message from the International Space Station. As an astronaut of the European Space Agency, I'm proud to be an ambassador for the Mission X 2018 challenge. During this challenge, you will be doing both physical exercises to keep your body fit and trying out science and technology experiments. It can be really fun to learn new things, and it will broaden your choices and give you new perspectives for your future. Here in space, we are free falling around Earth, and as a result, our body does not feel the pull of gravity. Without exercise, our muscle and skeleton degrade rapidly. Therefore, Not only we exercise about 2 hours per day, but we really pay attention to what we eat and drink. In fact, we have a team of doctors, nutritionists, and exercise specialists that guide us to keep our body in good shape. You too on Earth need to pay attention to your body, starting from doing exercising and sport activities, and pay attention to what you eat. During Mission X, you will learn about good nutrition, what food are best to eat when you are still growing, and learn how to work using scientifically correct methods. Well, we're going to have a lot of fun, and we will train together. You too can train like an astronaut. I wish you the very best of luck. I will be following your blogs. Go Mission X!
Okay, so a couple things I wanted to point out. If you get a copy of the resources, obviously with a copy of the PowerPoint slides, you'll get the links to the video that I just played, including the one that I played just before the program. Now, I'm seeing a couple people who are saying I can't hear, and I would like it if somebody could, or a couple people could let me know whether or not they can actually were able to hear the video or hear me, because I want to make sure that I'm broadcasting, because it could be a possibility of the connection on the other end. So it looks like, okay, so great, thank you. So for those of you who are having some technical difficulties, you may want to log in on a second device or log out and log back in. Sometimes it is an issue with the connection. So it seems as though, from what I'm seeing, is I'm broadcasting okay. Sometimes the videos come through and sometimes they don't. So if you're having trouble just with the videos, I apologize. There will be a link to them so that you'll be able to get the sound with it. So yeah, I've got the sound broadcast from my end. I've got the controls put up at maximum, so I'm broadcasting about as loud as I can. So you may want to check the volume on yours. The only problem is it may get really loud when I come back on if the video is really soft. So there's some ideas for figuring out how to troubleshoot if you're having problems with the sound. The one question that I had was about the launch for Mars 2020. They've actually pushed it back just a couple of minutes. It'll be 7:50 a.m. Eastern time. So I was pointed in there 7:54. That's what it was last week. It's now been pushed back a couple of minutes to 7:50 a.m. Eastern time on July 30th. So that way, just make sure I answered that question. And by the way, if I don't see your question right away, just keep asking. I will try to get the questions answered as I see them. So let's talk a little bit about the nervous system, just kind of the basics. So I will get my little green arrow up here. And so there's basically two types of cells when we talk about the nervous system in human beings. And that is, first off, the sort of star of the show is, as I always talk to my students, and that would be the neurons. These are the ones that are the ones that generate the signals. They are the ones that talk to each other. They talk to other organs in the body. And so, as I always describe it to my students, think of neurons as the celebrities. They are the stars. Everybody's attention is going to be on the neurons. And so there's three key pieces that I want to kind of point out in terms of how the cell is laid out. You've got the main body of the cell here, which has the nucleus. That's the cell body. In some cases, you've got some neurons that have these very bushy, short extensions on all sides of the cell body. Those are referred to as dendrites. That's the way the neuron receives information, whether it be from another cell, another neuron, or a receptor. So these are the inputs into a neuron. So these are the receiving. Think of them as like little satellite dishes. They're receiving information. And you got the cell body, and then you have one long extension that comes out from some part of the cell body, and that's referred to as the axon. And that is what's going to carry the electrical signal all the way from the cell body all the way to the very end to the tip here, which is called a synapse. And I'll talk a little bit about the synapse a little bit later. But I want to point out something about the axon. You'll see what looks like little blue ovals on the screen there. It kind of looks like a string of old-fashioned sausage. Those little blue pieces are pieces of protective covering called myelin. Some neurons have myelin, some do not. And if a neuron has myelin, it helps speed up the rate at which a neuron can send an electrical signal to another. So those are some key features: the receiving end, which is the dendrite, the main body, which is the cell body, and then one long extension, which is an axon, which has, in some cases, this myelin protective sheath that helps to speed up communication in some neurons that are in our brain. And then the other class of nervous system cells is what's referred to as neuroglia. And there are a couple of different types depending on whether you are in the central nervous system, which includes the brain and the spinal cord, or if you're in the peripheral nervous system, which is all the nerves outside of the brain and the spinal cord. And I won't go into each and every one of these. I basically, when I tell students, I mention I refer to the neurons as kind of the stars or the celebrities. I refer to the neuroglia as kind of the support people, the people who are hovering around the stars and taking care of all the various activities that need to be done to make sure that the celebrity shows up where they need to be and they're doing what they're supposed to be doing. Neuroglial do kind of the same thing with neurons. They help to keep the area around neurons clean. They help in some cases to provide that myelin sheath that helps to speed up conduction. Some of them even produce the cerebral spinal fluid that bathes the neurons and helps to make sure that the neurons function at their optimal levels. So the neuroglial are the second type of cell. Most of the attention, of course, goes to the neurons, but neurons wouldn't be able to function without the neuroglia. And so just basically taking a look at the nervous system, I've got two diagrams here. The one on the left is sort of a schematic. It shows you a couple different regions. The top here, which includes the brain and then the spinal cord, is what's referred to as the central nervous system, or the CNS. And then you have everything that's outside these nerves here, nerves in the hips, the torsos, and the legs. That's referred to as the peripheral nervous system, or the PNS. One branch of the peripheral nervous system is a sub-branch that innervates a lot of our organs, so things like the heart, things like the digestive system, things like the endocrine system. The autonomic nervous system is a part of that peripheral nervous system, and the autonomic nervous system, as its name kind of implies, helps to take care of sort of those automatic functions. So for example, beating of the heart, that is something that the autonomic nervous system oversees. And as I mentioned, I think maybe it was last seminar, if I had to remember for my heart to beat, I'd be in trouble because I'm a very forgetful person. So autonomic nervous system is that part of the peripheral nervous system that basically tells the body organs to do various things at various times. Okay. And so that's, and then sort of on the right side here, sorry, almost right, this is part of a really amazing exhibit that made its rounds around the world a couple of years ago. It was basically showing the human body, and it showed various ways of various systems that you find in the body. So this one is essentially taking a look at that nervous system, the brain, the spinal cord, and all of the nerves that are associated with our body. So you can kind of see how extensive this system is. And when you think about what it does, it actually kind of makes sense that something that's basically responsible for all the functioning of our cells inside of our bodies would be this extensive and widespread in our bodies. Okay. All right, so what do our nerves do for us? What does our nervous system do for us? Well, three main functions. So the first function would be the sensory, or sometimes what we refer to as afferent with an A. The sensory portion of the nervous system is basically collecting information. It's collecting data about things in the external environment. It's collecting data about the internal environment. So, for example, you're looking to cross the street. The nervous system is helping to collect information about, sounds and sights and things that you would need to be able to determine whether or not it's actually safe to cross the street at that point. Internally, that might be keeping track of the blood pressure or the heart rate or your respiration rate. How fast or slow are you breathing? And so the reason it's referred to as afferent is sensory information is always going towards the brain or towards the spinal cord. So that's one thing that this nervous system does for us. The second one is sort of that integration or processing. So in order to make a decision as to whether or not the body needs to respond to something, there needs to be some sort of central processing unit, kind of like a computer processing data. That typically ends up being what's referred to as interneurons, which are found in the spinal cord and the brain. And so depending on the information and the level of need for higher brain areas to get involved, sometimes the integration is done solely at the level of the spinal cord. Think something like a reflex, like pulling your hand off of a hot stove, or it can involve higher levels of perception and consciousness, which means it has to get up to the upper areas of the cortex, the brain, the cerebral cortex. So that's the second thing, that it has to be some way of making a decision. I've got this information coming in. Do I need to make a response? Yes or no? And once again, that can be a response internally or a response externally. And then the third and final major function that our nerves do for us is motor, or sometimes what's referred to as efferent with an E. And basically the motor part is action. If the sensory information is processed by the integration center and it's like, yep, we need to do something in response, it is the motor part of the nervous system that's its responsibility to actually execute through an effector to actually carry out that command. So if the spinal cord or the brain decides that an action needs to be made, that's going to be done through an effector. Now that could be, like in the case of pulling your hand off of the hot stove, that may be the skeletal muscles that are in your arm and your hand to pull that away. But it can also be an endocrine gland. So if you are in a stressful type of situation, and this goes back to some of the research that I used to do when I was a research scientist, that parts of your brain are going to send signals to your adrenal glands, which produce cortisol. Cortisol is released in response to stress. Epinephrine and norepinephrine is also released in response to stress. So it doesn't necessarily have to be a skeletal muscle to effect a response. It can be an endocrine gland. And the reason we call it efferent with an E is because the information is coming down from the brain and away from the spinal cord towards various parts of the body, which are actually going to carry out the action. All right, so time for a little interaction here. So if you were to take all the nerves or neurons in a human body, and you kind of saw that in that picture a couple slides, but how long do you think it would be? Lined them up end to end. Okay, a lot of miles, 3 miles, 12 meters, 250 miles. Okay. 50 meters, 2 miles. Okay. Got one with a football field. Okay. Encircle the Earth. Diana, you are the closest. Answer is, if we were to take all the nerves and line them up, it would be about 90,000 miles. And so if you take a comparison, for comparison purposes, the Earth's circumference is just under 25,000 miles. So not only is it one time around the Earth, Diana, it's actually almost 4 times. It's like 3.5, 3.25 times. That's if we were to take all the neurons, all the nerves, and line them up end to end in just a single string. And so I'm getting some questions. Really, yes, I can provide the citation for that in the resource. But yeah, it's It's quite a bit of length. Okay, so let's go from length to speed. So how fast can electrical signal travel? Okay, 1 millisecond. Okay, millisecond. Speed of light. Speed of light. Fast. Pico. Yeah. Okay, so yes, fast. This one actually is a little bit slower. So if we think, so let's see, PM, yeah, a little bit fast. So fast, yes. So maybe not as fast as the speed of light, or faster than the speed of light, but certainly fast. So most neurons will conduct at about 100 meters per second. Of course, we don't think that because our bodies are not 100 meters long. But if you take the velocity, it's 100 meters per second. And for those folks who work on the English SI system, that would be perhaps as fast as 180 miles per hour. So definitely faster than any cars that we have, even sports cars, even high performance sports car. I think you have to get to like Formula One or NASCAR before you get cars that are going a speed that's roughly approximate to the speed at which our nerves can conduct. Okay, so let's ask, can nerves regenerate themselves after injury? And I think I'm with Kathleen. I think sometimes my nerves are not that fast. Okay. All right. So the answer is you all are both right. Those of you who are saying yes, that is true. Those of you who are saying no, that is correct too. It really depends on where you're at in the nervous system. If you are in the brain or the spinal cord, chances are no, you're not going to be able to regenerate after a spinal cord injury. Think, for example, Christopher Reeve, Superman. He had a spinal cord injury, broke his horse. He had a horse riding accident. He's thrown from his horse and unfortunately broke his neck at the level, I think it was like C3, C4 in the neck, pretty high up, and he was bound to a wheelchair the rest of his life. And he made it his mission to actually bring awareness to spinal cord injury research. And the interesting thing about that is partly because of his efforts in bringing awareness to it. We have some individuals who now do actually sustain spinal cord injury who are actually able to walk. I'm originally an alumni of Penn State University. We had a fellow on our team in the late 90s. His name was Adam Talaferro. He broke his neck during a football game. And it was the type of injury that it was the same type of injury that Christopher Reeve had. They didn't think he was ever going to be able to walk again. But because he was nearby a hospital, in fact, one of the world premier hospitals in terms of spinal injury research, they managed to get him some steroids to reduce the swelling in his brain and particularly his spinal cord within an hour. If you can administer steroids within an hour, you prevent a lot of nerve damage from the swelling that happens as a result. And I'm happy to say that Adam, after about 8 months of therapy treatment, he was able to lead our team out onto the football field walking. He's a good friend of mine, and he doesn't have quite the control that normal people would have. But getting him those steroids in the first hour of the injury was so important to him being able to be able to walk. But in most cases, if you do damage to the central nervous system neurons, no, they're not going to regenerate. But peripheral nervous system, yes, peripheral nervous system has a wonderful capability of regenerating, and it has to do with some of those neuroglia and being able to regenerate those protective myelin sheaths that they are responsible for. So yeah, it is. I didn't know about the graphene, but that is a very promising area of research. And Dudan is asking, can nerves regenerate with mother cells? I think you're responding to stem cells. And there's lots of research Not sure if it's being done in the United States, but there is research being done at various labs around the world to basically take a look at the possibility of using stem cells as possible treatment. Yes. Okay, so which has more? Brain, and that's including everything, the neurons, the synapses, the neuroglia, or the stars? And I'm seeing the vast majority of people basically saying, let's go with the brain, because of course this is a program about the brain. And so the answer is yes, it is the brain. So particularly if you take a look at the connections, the synapses between the various neurons, yes, there are way more connections in our brain, even if you account for the fact that we lose neurons throughout the course of our life. Yes, we do have more cells, more connections, more neuroglia in our brains than there are stars in our Milky Way galaxy. And so there's lots of great stuff out there about the sort of similar architecture between our brains and stars in our Milky Way galaxy. But I will definitely make sure that I send out the website where I got those facts for you, so that you can check me up on them. And it says, why do we lose neurons? That's an interesting question, No Marie. So during fetal development, we end up producing loads more neurons. And depending on use or disuse, that is how you can lose them. So I think the statistic is a newborn can lose neurons just as a matter of development, and that's why they say it's so important to provide newborns and children between the ages of 0 and 5 that mental stimulation, because that helps to basically set the child for life in terms of the number of neurons that they're going to have. Neurons, once they specialize, they lose the ability to divide. And so they are so specialized that if we lose them, we can't replace them. Now, as was pointed out, if stem cell therapies get figured out and become a fairly widespread treatment, that may change. But for now, we don't necessarily have any way of replacing neurons once we lose them. So that's why they're always saying, not only for newborns, but then also for adults as we age, to make sure that we engage in mental stimulation, crossword puzzles, learning new things, stuff like that, that adults who are continue to regularly engage in intellectual activity have some protection against things like Alzheimer's and Parkinson's disease and memory loss, which I'll talk about a little bit later in this particular webinar. But that's essentially, we lose neurons either because we don't use them and the brain's like, oh, okay, well, we're not going to maintain things that we're not using, or just as a matter of aging process. So I want to bring your attention to a second resource for the teachers. This is a curriculum that's entitled The Brain in Space. This was tied in and released with the Neural Lab Space Shuttle program, Space Shuttle mission in 1998, which I'll talk a little bit about in a few slides. But the curriculum, the original one, is found at the web address that you see there. It's designed for grades 5 through 12. And it has a number of units that correspond to the different types of experiments that were done on that particular Neural Lab shuttle mission. You're very welcome, No Marie. And so they have various activities that help students to understand things like motor skills development, or balance, or how does our bodies figure out where we are in three-dimensional space. Blood pressure changes that go from laying down to standing up, and how might that be affected if we don't have gravity in space, and certainly things like sleep and circadian rhythms. So as I mentioned, the original curriculum was released in 1998 in association with the Neural Lab shuttle mission. There is an updated version that the Aerospace Education Services Project, which is actually the predecessor to the contract that I'm on right now. You may or may not remember back in 2010 and 2011, NASA had this thing called Summer of Innovation. And so the AESP project took a number of curriculum and kind of refreshed or updated them. So I also provide a link to an updated version of at least 2 of the activities that were in the original Brain in Space curriculum. Those activities in the AESP version are geared more towards grades 7 through 9. Okay. All right, so last sort of couple little things about the nervous system. So the way that you have electrical signals. So remember, neurons have to communicate with each other through electricity. And it's not necessarily electricity in the way that we think of in terms of lights, turning on and off the lights or turning off our computers, which runs on electrons. Electricity in our bodies, what I refer to as biological electricity, runs on sodium ions, Na+, and potassium ions, K+. There's a few other ones that are involved, but for sake of simplicity, we'll just leave it as sodium plus and potassium plus. These two ions help to generate the signals that neurons use to basically transmit signals down the entire length of their axon. And so I've got sort of a diagram here that kind of shows you how this might start. So we have to, it's kind of like dominoes. You tip one domino, the rest of them will fall. So if you start an action potential at one end and you have enough voltage, enough oomph, enough power, that signal will then transmit down the entire length of the axon, as we kind of see in sort of a still shot. So it's here on the far left. Give it a little bit of time. It's now wound its way down about halfway through the axon, and then the last thing here where my green arrow is where it's made its way all the way down the axon. So it's much more complicated than that, but essentially it is the movement of sodium and potassium ions in and out of a neuron that generates this biological electrical signal. And so because you're using ions, sodium plus, K plus, so think like saline solution that you might get administered if you're in the hospital, it sets up something called an electrochemical gradient. And this is something that has to be constantly maintained by the brain and any neuron in the spinal cord or in the peripheral nervous system. So it requires a pumping action to basically force sodium to one side of the membrane and potassium on the other side to basically set this whole system up so that it can work. Once you send a signal, the neuron has to reset itself. So these potassium pumps are very important in terms of setting up the neuron so it can send a signal, and then cleaning up and resetting so that the neuron can set another signal. And so what ends up happening is that uses oxygen. And so your brain and your spinal cord, the nervous tissue in your body can use anywhere between 20% of the oxygen that's in your blood, that's if you're resting and not really thinking, up to 50% of the oxygen that's in your body if you're like really deep in thought, trying to learn something or thinking about something. So the brain and the spinal cord, the nervous tissue, takes a lot of oxygen. And so Stephen is, actually I should go back because I saw another question. It says, how does the brain determine high or low IQ? And that's actually not so much the brain. It has to, the capacity of the brain to do the tasks that are asked of you on the IQ test, that's set by the neurons and the connections and being able to process that information. The test itself is what helps to determine whether or not somebody has high IQ or low IQ. And I don't have as much familiarity with how the IQ test was created, so I'm afraid I can't necessarily answer that question beyond psychologists will give the IQ test. The individual will solve the problems or the little tasks that are asked, and then depending on how many they get right and depending on how fast they're able to get those solutions, that is what the psychologists then use to determine what approximately someone's IQ is. So it's not so much the brain determining it as these measures that psychologists have basically decided upon as valid measures of somebody's cognitive or thinking capabilities. Steve Penderson is asking, if you have oxygen problems, does this affect the process? Yes. And I was actually going to, thank you, Stephen, I was actually going to make a point about COVID-19 because COVID-19, of course, affects the lungs. And if you affect the lungs' capacity to get oxygen into the blood, everything that depends on the blood for oxygen is by definition going to be affected. And so it's going to make it more difficult for the brain to be able to set up these neurons to basically create action potentials. So it might come across as like mental fog. If it's severe enough, you can start to lose cognitive or thinking capability. And if it gets serious enough, can actually lead to neuron death. And as mentioned before, neurons don't regenerate. So if we lose neurons, that can be a problem. And so the thing with COVID-19 is yeah, we might be able to handle what's going on in the lungs, the pneumonia, but one of the things that's being observed is that the oxygen levels in the blood are low, and that is catastrophic not only for other organs in the body, but especially for the brain. I think it's something like there's certain parts of our brain that if they don't get oxygen or they have reduced oxygen, within 5 minutes they start to die. So you can't have low oxygen for very long and expect to be cognitively all there. Okay, Robert, you're welcome. And Dudan is actually making a comment to fellow educators. There's an analogy to RC circuits used in physics. So yes, definitely there's a tie-in here with electricity and stuff like that. As I'd mentioned before, this is obviously not electricity like we think of with electrons and movement of electrons, but the ideas and the concepts are very similar. All right, so let's talk a little bit about actual space microgravity research. So I mentioned previously in this series that there is, so Henry is asking, does the brain consume a lot of energy compared to the rest of the body? Yes, because the brain has to basically maintain those gradients, those sodium and potassium gradients that make it possible to communicate with electricity. Those are constantly going. They constantly have to produce energy to force the sodium and potassium into the right places. It's called active transport, to force things into place so that the neurons are able to communicate. And so that's like constantly bailing water out of a sinking boat. So the brain is always expending energy to make sure that the neurons are in a state of readiness to send a signal when they need to. So yes, it ends up consuming a lot of energy resources. And I forget what the statistic is about the amount of food that you take in. Of course, that's converted to energy by our bodies. There's a fairly high percentage of whatever you eat is going to be used by the brain and the nervous tissue. So yes, huge energy input, and that must continue so long as the individual is alive. Okay, so I mentioned earlier in this series that there's been a couple of space life science missions. Those were sort of general purpose space life sciences. So they took a look at skeletal system, they took a look at muscles, they took a look at endocrine, they took a look at kidneys. There was one shuttle mission that was entirely devoted to the nervous system, and this was this was NASA's contribution to what became known as the Decade of the Brain. So from about 1998 to about 2008, scientists were really focused on understanding how our brains work, and so this was NASA's contribution, a 2-week shuttle mission that used the ESA Space Lab, launched in 1998 aboard Space Shuttle Columbia, just like the other 2 Space Lab missions that I mentioned previous. This ended up being the last scheduled flight of the Space Lab module, but it was a partnership. The experiments that were flown was a partnership not only between NASA and ESA, but also the French Space Agency, the German Space Agency, and the Japanese Space Agency. So all of these partners came together to determine the experiments that used a wide variety of test subjects. Not only were the crew members used, but we had rodents, we had insects, crickets. That must have been interesting to have crickets on the shuttle in their little habitats, snails, and then fish. And so, as I said, wide variety of experiments. The interesting thing about this particular shuttle mission, I mentioned earlier that I'm a Penn State grad. I was actually at Penn State. I was actually 3 weeks away from graduating when this mission went off. And one of our professors was actually a payload specialist on the shuttle mission. So you can see Dr. Pawalczyk here with the Penn State flag, which of course lots of astronauts do. They take flags up for their alma maters or the various branches of the service that they serve with. And so it was actually a really cool kind of tie-in, because of course I had been interested in the brain. I had presented on space life science in some of my physiology courses, and then to have a professor actually be a payload specialist on the flight was really kind of cool. And so if you're interested, particularly my university professors and my university students, I've pointed this out on the other ones, but I'll point it out again in terms of Neural Lab. There is a repository that will tell you about all those experiments that were done on the Neural Lab mission. I give you the general web address. This is for the entire repository of life science research that's been done in space, but the link that's specific to Neurolab and STS, that should have been STS, not STE, should have been STS 90, is that second link where my green arrow is right now. So they give you a summary of the mission, they give you a picture gallery, and then they list out the specific experiments, so you can click on those links to learn more about each individual experiment that was done on Neurolab. And they had like a whole symposium where they came back and they talked about the results and stuff like that. So what I'm going to do is I'm actually, because I'm running out of time, it's 6:45, I'm actually going to skip over this particular video, but I do have it. Obviously, you'll get it if you get the resource link. So let me go in between Neurolab and ISS and bed rest studies. This is where we're getting the data that tells us what happens to the nervous system when the body is in space. So I've pointed this out before, so I'm going to skip over that. I'm going to talk a little bit about now some of the ISS research. And so trying to find all of the entries for all of the life science and particularly nervous system research is a little bit tricky because it's done over the course of several expeditions. So I just had to randomly pick one. I ended up picking Expedition 42, which was right before the NASA Twin study, and it has at least 3 different experiments that were done over the course of a long period of time, a couple of years, that deal with the nervous system. So one, for example, took a look at how does fatigue and insomnia affect cognition. Also fluid shift, when we don't have gravity pulling the blood down towards the lower part of our body, that pools up here in our face and the upper torso. What does that do to the intracranial pressure inside of our skull? And as it turns out, that fluid shift increases the pressure in the skull and can actually lead to visual impairments. Astronauts, particularly male astronauts, are reporting visual problems in space and also when they come back down. And then there's another one that actually deals with just blood flow. So I was talking about how fluids shift and tend to pool in the head and the upper torso. There's a third experiment that was done on Expedition 32 that was looking at, okay, how does the brain actually drain the stuff? Because in gravity, we normally have gravity to pull the blood down towards the heart. We don't have that in space. So how does the brain actually, is it capable of draining the fluid, whether it be blood or cerebral spinal fluid, down out of the head and down into other parts of the body? Okay, so let's see, how many experiments are done at the ISS at once? Dorothy, I don't actually have an actual number for you. It is many, many. And it's not just life sciences. There's material science, there's physics, there's engineering, there's technology. So, I mean, and that's also not including what's done inside. There are some external palettes that are doing, that are collecting data as well. So I would say that there's probably 10, 20, probably more like 50, 60, probably almost 100 experiments being done on the space station at any given time. Not all of them require astronaut oversight. So some student programs will send experiments up, something called nanoracks. Those experiments are entirely self-sufficient. The astronauts don't have to do anything with them. So I don't actually have an actual number for you, Michelle. Is it harder to work out in the ISS than it is on Earth? And the answer to that is yes. So I was going to quick kind of point out that in order for one neuron to, and I'll get to that in just a second, because this piece with the neurotransmitters and the synapse actually plays into the research that's kind of helping us decide what is going on with the human brain. But the short answer to your question, Michelle, is it is harder to work on the ISS. There are very distinct impairments to our ability to do things both thinking-wise and like motor skills-wise. That's the short answer. So with the neurotransmitters, you have the neuron, which has that electrical signal, but once it gets to the end of the axon at the synapse, in order for it to communicate between neuron 1, which is where my green arrow is, and neuron 2, which is where I've now moved the green arrow to, there has to be some chemical messenger that crosses the physical divide. So no neuron actually physically touches another one. There's a small space in between the two. And so that's where neurotransmitters, these little chemical messengers like serotonin, dopamine, etc., come into play. And a lot of diseases that affect the nervous system appear to be the result of some malfunction associated with the neurotransmitter. Okay, and so just for sort of two examples of diseases that are being looked at on the ISS with the hope of understanding them in terms of what actually is going on in the ISS and the unique environment of microgravity, being able to understand what's going on for Earth-based medicine applications is two of these, Alzheimer's disease and Parkinson's disease. So Alzheimer's disease results in eventually dementia, memory loss, also ability to lose control of motor functions. And when we take a look at the actual brain tissue, it actually is reduced in patients who have severe Alzheimer's. In the case of Parkinson's disease, I bring you to the area which is red in a healthy individual. This is the result of a loss of neurotransmitters, so that over time, this individual who has Parkinson's disease loses the ability of those neurons to speak to other neurons, and that's where we start to get the trembling that we associate with Parkinson's disease. Okay. So those are two diseases among many that are of particular interest. I've got another video, which for sake of time I'm going to skip, but it is a protein crystallization experiment. Down here on Earth, if we want to understand the structure of a protein, or in the case of DNA, we usually make crystals out of the material. But because of gravity and other issues being down here on the Earth, lots of impurities can get into the crystals, and so it can make it difficult to figure out the three-dimensional structure of a particular protein or a three-dimensional structure like DNA. But it turns out if we go into microgravity, we can grow pure, less contaminated crystals that help doctors and scientists characterize or determine the three-dimensional structure of, let's say, a protein much more quickly. And so there's currently a study on the ISS. It's called the LRRK2 protein crystallization. LRRK2 is a protein that has been associated with symptoms of Parkinson's disease, but nobody has been able to figure out the 3D structure by making crystals of it down here. And so now up on the ISS, they're trying to make crystals of LRRK2 to help figure out the three-dimensional structure. And if we know the three-dimensional structure, we can figure out drug therapies and hopefully help treat Parkinson's disease down here on Earth. Okay. Is dyslexia considered a neurological issue? Off the top of my head, Tanya, I'm not certain. But I'm going to say yes. I have auditory dyslexia, and that is the result of auditory nerves in my auditory cortex not having necessarily made the right connections. So for regular, regular dyslexia, the flipping of the letters, flipping of the numbers, yes, there is something happening with the neurons and how they've made connections with each other, or how they're communicating or not communicating, however you want to look at it, in basically the underlying issue, yes. Okay. I'm seeing some comments here, not sure what Laura, are you asking a question? I'm not sure I understand all the little symbols there, so I'm not sure what's going on there. But I want to point out another thing that's happening on the ISS is coming up with models. So for us to understand how a neurological disease onsets, okay, that's what I was thinking, Laura. I wasn't sure, but I want to double check. So if we want to understand the onset of a neurological disease or its progression, we have a problem. We can make animal models, which with rodents, they're very similar to us, so it's okay. But if we really want to understand how it works in a human, we have a problem. We can't just go doing research on normal, healthy human individuals for ethical considerations. And so trying to figure out, okay, how do we get around the fact that rodents aren't exactly like us and the ethics of doing human research, what one idea is, what's being tested is, can we reprogram stem cells to basically create regions of the brain, which are called organoids. So they're not a full brain, but they're like a portion of the brain. And so they're living masses. They make connections just like normal brain tissue. They organize into structures that we could readily recognize as, oh yeah, that is similar to what that area is in the brain. And by being able to do that, now we have something that is fairly similar to the real thing, and so we can actually test things. We can understand better how the disease progresses, and then if we can figure out ways to treat it, now we have a way of treating it without actually having to treat it on a human being. So it kind of helps in this process of better characterizing and treating neurological diseases. And so this particular study that I referenced here is actually looking at the progression, the development and the progression of both Parkinson's disease, which I showed you earlier, and also primary progressive multiple sclerosis. So hopefully that will work, and we'll be able to have that as a resource down here on Earth. All right, so I got about 5 minutes. Okay, I think I've only got about 2 more slides here. So one of the things, and this goes to a question that I think Michelle was asking earlier, is it harder to work on the ISS than it is in Earth? And the answer that I said to that was yes. Astronauts for a long time have been reporting the fact that it's harder for them to control their physical movements, and they almost have like a little bit of a brain fog. They find that it's much harder to concentrate and complete mental tasks. They also report problems with balance, particularly when they come back down to Earth. But when they're up in space, of course, there is really no such thing as up or down, but the brain is frantically trying to figure out what is up or down. And so there's some perceptual issues that the astronauts are reporting where one minute they think, okay, they're right side up, and then the next minute it's like they feel like they're upside down. And it's really hard to describe because we here down on Earth can't really, imagine, okay, having that issue of what is up and what is down. We know what up and what is down, but when you're up in space, that's all gone. So this was a study, I think it's actually ongoing, it's called neuromapping, and what they're doing is they're doing functional MRIs. So these are living tissues. They're basically just doing scans of the astronauts' brains before and after they come back from space. The behavioral assessments, things like obstacle courses or spatial memory tests, those will be done before, during, and after spaceflight. And so basically what they're looking for, what the researchers were looking for, is are there any physical changes that we can see in the MRI? Do certain areas of the brain get smaller or larger? Can we see that? And does that actually revert once the astronaut has come back? So when the astronaut first lands, they take an MRI, and then about 6 months later they take another MRI to see, okay, has the astronaut's brain areas come back to normal? And what they find is that yes, brain area size changes, and this is something that's referred to as neuroplasticity. We don't necessarily think of the brain as being a dynamic structure that kind of reorganizes how it handles various tasks, because we don't think that neurons actually regenerate, but you can to some extent change connections between neurons. You can strengthen neurons or strengthen connections. You can lessen connections. So there's some things that the brain can do to adapt even if it can't make more neurons like in bulk. And that's what neuroplasticity is. It's sort of changing the ability of the brain to actually handle certain things by sort of reorganizing the connections. And that's what makes the brain areas look bigger or smaller, is the reorganization of those connections. And one of the interesting things kind of tied in with the fluid shift that I mentioned, the brain not being able to get that fluid down, the cerebrospinal fluid down, forces the ventricles inside the brain, which normally are the repositories of cerebrospinal fluid, it forces them to become bigger. So this last point that I have here down where the green arrow is they're seeing some significant increases in the volume of the ventricles, those open, what look like open pockets in the brain. It's where the cerebrospinal fluid flows through the brain as it's going down to the spinal cord. And in astronauts who were up for 12 months, so that would include Scott Kelly, the ventricle increase was much bigger than those increases that were seen in astronauts were just there for 6 months. So the longer the astronauts are in space, the greater the change. Now, in most cases, the changes in brain areas, they went back to pre-flight, but interestingly enough, the ventricle volume did not go back 6 months post-flight. So that may be a more permanent change, and that may explain to some extent why astronauts are having issues being able to control their physical movements, being able to do these mental tasks, having that brain fog. So it's very definite that there are brain area changes. Okay. 2 more slides. This is another instructional resource. This is what's called Learning Launchers. They are little toolkits that include videos, activities, and curriculum. They go for a number of different areas, so physical and life sciences, material science, etc. They're geared for grades K through 12 and to particular interest with this particular topic, nervous system. There were 3 learning launchers, one called We've Got Rhythm that takes a look at circadian rhythms when we're awake, when we're asleep. Another one is tied in with the curriculum I pointed out earlier, the brain and space. And then the third one is taking a closer look at this visual impairment, this problem that the male astronauts in particular are having in terms of their vision and being able to see not only while they're in space, but then also when they come back. Now, the interesting thing with the visual impairment is it does seem to resolve itself with time after the astronaut comes back, and that may be, once again, that fluid shift. We're seeing the ventricles get enlarged. That may be putting pressure on the optic nerve, which is creating the situation where it makes it harder for the astronauts to see. Emily is asking, are dreams different in space? I don't know for a fact whether or not that is true or not, Emily. I'm going to say tentatively yes, even though I don't have any evidence to suggest one way or the other. I'm not even sure, although I'm assuming that they have done dream studies. I know that there have been at least a couple studies taking a look at REM, the amount of REM sleep that the astronauts get. I think that goes back to like shuttle missions. But I don't know that anybody has actually done the follow-up work to see, okay, does an astronaut dream more vividly in space or not? Do they even have dreams at all? I'm not sure entirely what the answer to that question is. How about the question about spatial orientation? I think I missed that one, Michelle. So let me see if I can quick scroll back through and see if I can find, actually, I'm not sure question. So spatial orientation, you might have to help me, Michelle. What was your question? I'm not seeing it real quick. So while I'm waiting for Michelle to type in her question, and you're welcome, Emily, so here are some possible countermeasures. We still have a lot to understand about what long-duration spaceflight does in terms of just effects. So trying to determine countermeasures is a bit of a work in progress at this point. The more that we come to understand how long-duration spaceflight affects us and our nervous system function, the more developed that these countermeasures are going to be. And so, as you might imagine, this is a challenge with going to Mars. And so these sorts of things have to be solved before we can actually attempt. But it doesn't mean that we can't solve them. It just means that they're ongoing. We're learning using the International Space Station to figure out what is going on. And once we figure out what's going on, then we can start trying different countermeasures to address that and hopefully find ones that will be successful. And so Michelle is asking, does looking at planet Earth help with spatial orientation? To some extent, yes, if they're at the cupola, and there is Miss Ninja's video bomb for the program. Okay, and so taking a look, if you're in a portion of the space station where you can actually see the Earth, that's great. But if you've seen pictures of the various modules, not all of the modules in the International Space Station have windows like that. And so if they're in a portion of the space station which doesn't really have a window out for them to see the planet Earth, that may not necessarily help them. It may only help them when they're actually in the cupola, which is also a portion that was contributed by the Italian Space Agency. So it can, but the degree to which it can help kind of varies because the astronauts are not always in the area of the cupola or in an area where they might be able to see outside. So here are some suggested countermeasures that are in current stages of testing to see whether or not they help. Motor imagery is a technique that's used in sports. It's also used in pain management and rehabilitation, where the individual is mentally thinking about the various muscles that are used and needed. It's kind of like a muscle training visualization technique. And so this is something that's being tested as the astronaut using this visualization technique to basically get themselves or train their minds to be able to do the task more readily than if they just try to do it. So it's a technique that's, as I said, it's been used in sports, particularly among elite athletes, and so they're testing to see whether or not this might be helpful. Another one, which is a picture I have in the lower left corner here, is transcranial magnetic stimulation. You may have seen in the first slide there was like a little cap that was on that had like little electrodes, or you just have a coil that generates a magnetic field, and just by stimulation through the scalp of motor cortex, that can help stimulate muscle contractions, which of course in microgravity we don't necessarily use our muscles all that often, and so this can be a way to kind of help maybe offset some of the issues with the brain communicating with the legs that doesn't get worked as much when we're in microgravity. Some things like pharmacological or drug interventions are kind of too early to tell. The only one that's kind of well known would be the motion sickness. There's several different drugs that astronauts, and of course we down here on Earth, can take to help alleviate the symptoms of motion sickness. And one other one, which is more of a psychological and mood, might be journaling. Astronauts keep extensive journals while they're up in space, and that can help cope with coping with isolation and confinement. It also gives folks who design spacecraft insight into, all right, how might we want to design a spacecraft that's going to Mars where the astronauts will be confined to it for possibly 3 years or so. So as I said, these are in various stages of testing and development. We don't really have as well developed countermeasures for nervous system impairment as we do for skeletal or muscular, but that's simply because the brain is a very complex organ and we're still learning about it. But there's multiple benefits, not only in terms of us going to Mars to be able to protect our astronauts, but also the benefits of helping us understand just healthy aging and other neurological disorders is just immeasurable. And so that's what the International Space Station does for us. So I apologize for running over by a couple of minutes. There is my email address. If you have not already done so, please put your email in the chat box and note if you'd like the resource email. The ones for muscles will go out tomorrow. I'll try to get the one for this one out tomorrow as well. Post-event surveys will go out within the next 24 hours, so sometime July 24th you'll get that email with the link to complete the survey to grade me. In a week, which will be July 30th, you'll get your certificate email with the link so you can print out your certificate for you teachers and students. I've recorded this as well. I also give you the next couple of webinars. I will point out that the next two may or may not appear on the webinar list. So I have included the registration links for my next two webinars. So next Thursday, I'll finish off this series, Human Factors, with taking a look at genetic changes in the DNA that happen as a result of long-duration spaceflight. That will particularly focus on what has happened to Scott Kelly as a result of the year mission. And then 2 weeks from tonight, I'll start a new series focusing on things that Langley Research Center, my home center, focuses on. We'll take a look at fire safety during this wildfire season. So thank you very, very much. For those of you who just give me your email, if you want resources, just type in you want resources so that I can put you on the list. Otherwise, please stay safe. I will stay on for a few minutes if you have questions. I always answer questions afterwards. So if I missed one during the program, please feel free to let me know. Otherwise, thank you and have a wonderful day wherever you are in the world, and stay safe. And I hope to see you next Thursday for the series finale when we talk about DNA. And Miss Ninja is about to say hello to everybody. So for those of you who don't know Miss Ninja, this is NASA's feline education specialist. She is a 15-and-a-half-year-old calico tabby, and she makes it a habit to video bomb our webinars at least once, if not, as tonight, twice. Sometimes she even does it 3 times. But Miss Ninja says hello to everyone and purrs her appreciation. So thank you very much. Susan, I see that. I'll make sure you get a copy of the I already sent the skeletal system out, so I'll send you a copy of that one. I haven't sent the muscular one out, so I'll make sure to put you on the list. That muscular one should go out tomorrow. I'm a little bit behind. So yes, I can still get those resources to you, Susan. Robert, have a great evening if you're still with me. I'm glad you enjoyed it. Kathleen, you're welcome. Virginia, you're welcome. Okay. Emily, you're very welcome. Okay, Mina, I know you put your email in there. I'll make sure you get the resources too. Okay. All right, I'm looking for questions here. If you got questions, fire away. Well, maybe not fire. Well, Neuron's fire. So yeah, go ahead, fire away if you have questions. Okay. Ella, you're welcome. Mark, you're welcome. Caden, will do. I'll send you the resources. Naomi, you're welcome. I will do. Okay. Francisco, I will do so. Yeah, Larissa, yes. Miss Ninja's a cutie pie. Up. I think we're going to get a third one here. Yep, there she goes. Photo bomb number 3. Or video bomb number 3, I should say. Okay. Dudan, you're welcome. Okay. Patty, you're very welcome. Aaron, you're very welcome. Yes, you all have a nice weekend. Take care. Julio, Julio, we'll see you next week. Ekta, you're welcome. Let's see, I'm seeing some, okay, emails. Lunchtime. Anj, if you are still here, if it's 7:50 Eastern time, that will make it 4:50 Pacific time. So Anj, if you're still here, and I hope I'm pronouncing your name correctly, it will be 7:50 Eastern time, 4:50 Pacific time for the Mars 2020 Perseverance launch next week. Okay. Okay, Rosetta, you're asking what is the link for the wrench that was made through the 3D printer? So hold on just a second. NASA. See if I can find that real quick. All right, so here's the link to the story, Rosetta, if you're still here. Okay. Go back here. So if you're still with us, Rosetta, I just put the link in. Aaron, okay, noted. I will make sure you get the resources. Okay, Stephen, you're welcome. I'll be looking for the email. You too, Stephen. Maria, you're welcome. Okay, Aaron, yep, got your email. Okay, Veritas, you're very welcome. Yep, Miss Ninja's happy. Miss Ninja's happy too. You've been waiting all day to make her appearance. Shahida, I see that you're going to need both, actually all 3, the skeletal, the muscular, and the brain ones. Okay, sounds good. Okay. Navarro, I'm seeing parentheses after your email, so I'm going to assume that you want the resources. BP, got it? Okay. Okay, Maria, yep, got the resources. You're very welcome. Okay, Marites, I'm glad. Looking forward to having everybody back next week. Mark, yes. Yes, and I think I have your email somewhere, Mark, so yeah. So the fact that I'm seeing you put a note there, yes, I can send you a copy of the slide so you can at least see the well, you'll get the resources for everything, for both of those webinars. Okay. You're welcome, Rosetta. You're welcome, Matthew. Okay. Yeah, I think I did see that you attended, Dawn, but I may have missed that you wanted the resources for all three. So I'll assume, Dawn, that you want the resources for all three. Unless you tell me otherwise, Dawn, but I'll assume that you want all three. See you, Dudan. You're welcome, Mark. Okay. Okay, cool. Yeah, so I'll just assume, Dawn, that if you haven't already gotten the others, I'll put you on. Matthew, it looks like you want all three as well. Okay, Kiyomi. Hopefully, I hope I'm pronouncing your name correctly, Kiyomi. Rosetta, you should be able to register for it now for the July 30th. I don't know if you tried to register earlier. It was down for a bit, but it's back up. That's why I put the web address there on the screen. So if you go to the web address that's listed for next Thursdays, you should be able to register for it. Okay, Kiyomi. Whew. I always worry. Like, did I pronounce it correctly? So, okay, cool. Okay, Rosetta, you're welcome. Sounds good, Mark. Thank you, Adrian. Yeah, actually, the DNA one, so tonight's dealt with the brain, which, as I mentioned, I was trained as a neuroendocrinologist, so I very much love the brain. The DNA is my other love. That was, they'll tie in the genetics research that I did at Los Alamos. So tonight's and next week's I'm really super excited about because they're directly in my wheelhouse in terms of my training. So looking forward to it. And of course, it takes a look directly at what happened in the twin study and what happened with Scott Kelly and what now distinguishes him from his, they're no longer identical twins. So if you have any questions, please feel free to ask. I think I caught ones that were asked, but if I missed a question, please let me know. Said I'm going to stick around for another minute or two. It's getting to be feeding time for Miss Ninja and Mr. Puck. So I'll stick around for a couple more minutes if you have any questions. I'm going to stop the recording right now.